bit about a couple of things that can go wrong related to the stomach, and then move on to the pancreas. So I draw another stomach here, and the reason for this is when we're talking about motility, traditionally the stomach is divided into an roughly upper and lower region. We talked about three regions before: the fundus, the main body of the stomach, and the antral region, and those of course are still there. But in terms of what goes on in movement and mixing and so on, the upper portion of the stomach here is called the orad, or proximal part of the stomach. And the lower region is the distal, or caudate region. Orad meaning side closest to the mouth, caudate tail end of the body, proximal or distal. And these two regions have got quite different motility functions. The main function of the upper or proximal part of the stomach up here is to act as a reservoir, whereas the lower portion acts as a mixer, a grinder, and a pump. So what I want to do is just have a brief look at what's involved here. So first of all, the reservoir function. The stomach is able to expand a lot. I can't remember exact figures, but a resting stomach has got maybe tens of millimeters of capacity, and it can expand to hold, I don't know, maybe a litre or so of contents. And when the food comes from the esophagus and goes through the lower esophageal sphincter, it of course first goes into the upper portion of the stomach, and this part undergoes what is called receptive relaxation. Receptive relaxation allows for an increase in the volume without much significant change in pressure or tension in that region. And an analogy might be something like, you know, those suitcases that have got sort of folding bits. If you stuff more and more clothes, contents into that suitcase, the folded bits normally can expand, and the thing doesn't burst. If you just had a rigid suitcase and kept on putting more stuff in, then you can increase the pressure inside there. So the first part of the stomach is a bit like that. So once the food has been moved from the esophagus into the first part of the stomach, it doesn't undergo much excitement in the first maybe 30 to 60 minutes. It tends to sit in this upper part of the stomach and gradually get a little bit mixing with gastric juice, but it takes, as I say, maybe 30 to 60 minutes. And one of the significant points about that is that during that initial time in the stomach, the salivary enzyme amylase can continue to work. The pH optimum for salivary amylase is at neutral. So once mixing with the hydrochloric acid does take place, which of course it will do in the stomach, then salivary amylase is cut off. Its activity is stopped. But initially it can keep working. And then gradually, as more food is swallowed, the contents will move down into the lower part of the stomach, the distal part, where you get a much more active type of activity and more activity, and you get mixing of the food with the gastric juices, and you get a mixture called chyme, which is a mixture of the food plus the gastric juices, and this is mixed and ground to smaller particles, so you've got a mechanical breakdown here, and eventually this lower region will also pump. It will move the food on at the appropriate rate into the small intestine. Regulation of motility is extremely important because you have to make sure that the food or the chyme mixture moves just the right rate. Not too fast, because if it stomach in a big rush, it's going to overwhelm the functions which are largely digestive and absorptive of the small intestine. If it happens too slowly, then the next meal is going to come along and you get a sort of build-up and then, it's not going to work properly. So things have to go just at the right pace. So how is motility regulated, particularly in this lower mixing, grinding, pumping part of the stomach? This is shown in figure number 33. And remember that the stomach has got the usual layers of muscle. It's got those nerve plexuses, it's got pacemakers, it shows a slow wave activity or the basic electrical rhythm which we talked about earlier on. And the rate at which those slow waves occur in the stomach is approximately 3 per minute. And remember the significance of these pacemakers, they initiate the slow waves themselves then cause contraction, but if spikes or action potentials occur on top of the slow waves, then you do get contraction. And what is thought to happen is that roughly in the midpoint of the stomach, there's a sort of dominating pacemaker region, which initiates these basic waves, under the appropriate stimulation, you get action potentials, and you get waves of peristalsis. And these waves of peristalsis move down the lower part of the stomach at a maximum rate of about 3 per minute, based on the frequency of the slow waves. And the function of these peristaltic waves, it is fairly obvious and suggests that they're going to push the contents, push the chyme ahead of them towards the exit from the stomach and into the small intestine. So here's one wave, here's the second wave developing, following on from the first one, maybe another one, maybe 20 seconds later, pushing the food down. It's not quite that straightforward. These peristaltic waves become stronger and faster as they approach the exit from the stomach. And what happens sometimes is that the wave, the peristaltic wave, which is essentially a ring of contraction, will overtake the stomach contents because they're getting faster and faster as they go down there. And if that happens, the pyloric sphincter region here may be closed off on a temporary basis because of this peristaltic wave reaching the pyloric sphincter region. And if the food then comes trying to get out of the stomach, it can't, because the exit is closed off. And when you first hear about that, you think, well, that doesn't make sense. You're trying to get the food out of the stomach. If it closes off, it doesn't seem to make sense. Well, in fact, it does. What you get is what's called retropulsion. If the food is moving down towards the exit, and the exit is closed off, and another peristaltic wave is coming behind it, it can't go forwards, so it sort of moves backwards. And that's actually useful because it allows for further grinding, further mixing, and then another peristaltic wave will come down, and if it catches the right moment, it will find an open or relaxed sphincter and it will be able to go out. And the contents in the stomach are normally broken down to less than 1 millimeter before they are allowed to escape into the small intestine. So larger lumps of food or anything else that might have been swallowed will not normally be able to go out. Then that raises another question. People swallow coins. What happens? They don't stay in the stomach forever. Eventually they will be passed out of the body. How can it do that? I've just said that things have to be broken down to less than 1 millimeter before they can go into the small intestine. Well, that statement is true when you eat a meal. But what about what happens in between meals? And this is sort of a bit of an aside, but it's related to this. There's something called the migrating motility complex. migrating because it moves. Motility, because it's to do with contractions and relaxations, complex. This is a wave of contractions, peristaltic contractions, which occurs in between meals. So when you haven't eaten for a while, you've got rid of the meal you just had, it's a couple of hours, 2 or 3 hours before the next meal. Every so often, roughly every 1.5 to 2 hours, you get this complex, this MMC, which starts in the stomach, moves through into the small intestine, and all the way down. And its function is described as having a housekeeping function. So if there is anything left behind, it will be cleaned out by this type of activity which occurs between meals. So it's cleaning things up, getting it neat and tidy, and getting it all ready for the next meal. Now, how can these bigger things get through that sphincter? During this MMC, apparently the sphincter will open more than it does after you've eaten a meal. So it must be, I don't know all the details, but it must be some way of sensing what is present there and the timing, so what it is, the timing and so on, that allows for different degrees of relaxation of that allied sphincter. What causes this MMC is still not worked out all that well, but a reasonable suggestion is the hormone motility. And this was one of the 5 that I mentioned in the first lecture that was a traditional hormone of the GI tract. We don't know all that much about it. I won't be saying anything more about it, but this is maybe where it plays a role. Okay, so we've got the food then gradually leaving the stomach, and it goes into the small intestine. And we'll pick up on that again when we move into that region. Just a quick mention of a couple of things that can go wrong. There's never a good time to talk about vomiting. It's either just after you've had a meal or there's another one coming up. But here it is. So vomiting, or emesis, as it's also known, is the abnormal removal of stomach contents, and sometimes also the upper small intestine contents are moved in the wrong direction, upwards and out. So just briefly, what causes it and what's involved. It's regulated by a center, the aptly named vomiting center up in the medulla, and near to the vomiting center is something called a chemoreceptive trigger zone, also in the medulla, which is actually outside the blood-brain barrier, and so is able to respond to blood alcohol agents. So the sorts of things that can cause vomiting, and everybody probably has their own something to add to this, but there are things that we think about as, you know, the disgusting smell, or the nauseating sight, something you see something that really turns you off, and so on. So these can have input to the vomiting center. Problems within the GI tract itself, excessive distension, blockage, and so on, infections, motion sickness, pregnancy, particularly early pregnancy, so-called morning sickness, problems in the brain, increasing pressure and so on, and things that are in the blood, too much alcohol, various drugs, particularly things like chemotherapy. Drugs used in chemotherapy are notorious for causing nausea and vomiting. And these act through this trigger zone here. And I said, you probably add things to this. And here at the bottom, I won't go into a lot of detail. Here, this is what happens. Nausea is the sensation that something nasty is going to happen before it actually has. You get increased salivation, because if the stomach contents are going to be coming up, or the acid coming up, you need extra protection, bicarbonate and saliva. Changes in respiration. And the key thing really is that you have a relaxation of the lower esophageal sphincter at a time when the pressure on the stomach and maybe upper small intestine is increasing. So the abdominal muscles, not the stomach wall muscles, but the walls of the abdomen, these contract, pushing down on the stomach, and the contents can then move up through the lower esophageal sphincter. If the upper esophageal sphincter is closed, then the food content can't come out, then go down again, and this is retching, up and then down a few times before maybe eventually it does come out when the upper sphincter is open, and then various protective mechanisms for lungs and nasal passages and so on. The other problem is ulcers. So the next figure tells you some of the background to this, tells you what a peptic ulcer is, and it says a little bit about the causation and treatment. And I think the main reason for we mention this here is that, and I've got a couple, if anybody really wants to follow up on this, because it's a very interesting area. I have a couple of references here, I won't right now, but you can try and get them, which tells you more about some of the history of these treatments. And it ties in with, it's almost a revision of the physiology. So let me just explain what I mean. So it tells you what a peptic ulcer is. It's an eating away of part of the inner lining of the stomach or the small intestine or the esophagus. In general, an ulcer can happen anywhere, but peptic ulcers are related to the stomach and nearby areas. As it says here, the general cause is an imbalance between what's called aggressive and protective mediators or molecules or processes. So aggressive things would be hydrochloric acid, which we know is destructive. Somebody's asking about this, can it break down food itself? Yes, it can. It can denature proteins, and the proteins in the stomach wall. These can be acted on by hydrochloric acid if the protective side of this equation is not working properly. And then the enzyme pepsin, proteolytic, is going to eat away at proteins in the wall. The protective side, the mucus, the bicarbonate, normally prevent this, these aggressive factors from winning. But if you have too much aggression or not enough protection, then you've upset that balance. However, it used to be thought that it was just the acid that caused the ulcer. What people have now found out, that probably a major cause is a bacterial infection, Helicobacter pylori. It's probably a major cause. And this is a really interesting story. The Nobel Prize for discovering this was given in 2005 to a couple of workers, Warren and Marshall. And some of the work they did is something which you really shouldn't repeat at home. One of them, I think it was Marshall, actually deliberately got himself infected with this particular bacterium, Helicobacter pylori. He ate it or drank it or something like that. And yes, he developed problems and amongst a lot of other things, showed that this particular cause was extremely important. And it's said that, and he survived, he was okay, they managed to treat him and everything, but he was okay, but as I said, not something one should do oneself. Apparently about half the people in the world are infected with this bacterium, but not everybody gets the problem. Not everybody has ulcers. And there's still a lot to work out about it in regions where people are less well off, have less access to health services, have less access to safe water and food and so on, maybe not surprisingly, there's a higher incidence of this infection. In so-called developed areas, it's less so, but even in places like North America and Western Europe, I don't know the exact figure, but probably 30–40% or so of the population has got this infection. So what does it do? Well, this is one exception to the rule that bacteria are killed off in the stomach. This particular bacteria has got various tricks of its own which allows it to bury itself in that mucus layer lining the stomach. You can sort of corkscrew shape, it can work its way into that layer. It's got an enzyme, urease, which allows it to produce ammonia and get something else which creates a micro environment around this bacterium which is alkaline, and it can survive there. The body senses it somehow and it mounts an inflammatory response, but it's not usually a successful response because it's been hidden away. So it's thought that this inflammatory response caused by the presence of this bacterium is largely responsible for the problems here. Okay, and then there's some other things here. Now the treatment uses antibiotics and a proton pump inhibitor. And these proton pump inhibitors are relatively new, relatively successful, I believe also relatively expensive, and also recently there's been some questions as to their safety. What they do is to block the hydrogen potassium ATPase. So if you think back to how acid is secreted, you need that apical hydrogen potassium pump in order to push the hydrogen ions into the liquid. These PPIs, proton pump or hydrogen pump inhibitors, somehow bind to that ATPase and they block its activity. But only the ones on the cell surface, not the ones that are sequestered away inside the cell. So just the ones on the surface are blocked by these inhibitors. And a combination of using something to try and kill off the bacteria, preventing the activity of the hydrogen pump can be successful. And using this with some slight additions over a period of, I think, a couple of weeks, 2 or 3 weeks, it's been shown that you can get a very high, 90% or more success rate in treatment of the ulcer. If people get ulcers but they don't have the bacteria, then there's no point in using the antibiotics. You can try using proton pump inhibitors. Somewhat older thing was to give histamine blockers. And the interesting or important point here is that the histamine receptors in the stomach, and remember histamine is also a potent stimulator of gastric secretion, those histamine receptors are not the same type as the ones you find, for example, in inflammatory responses, where histamine can cause inflammation. Those are H1 receptors. These ones in the stomach, which cause gastric secretion, are H2. So you have to have special H2 blockers, which are available to prevent them. Prostaglandin-like drugs. Prostaglandins tend to be on the protective side of that equation. They probably work by increasing production of mucus, the bicarbonates, and the others too don't know that, and so they tend to be protective. Cutting the vagus nerve would work. The branches that go to the stomach is now obsolete because we've got these other ones. Okay, so to move on now into the small intestine, and the first secretion that I'm going to be talking about there is the one from the stomach. So we moved from the stomach through the thyroid sphincter into the duodenum, the first part of the small intestine, and this is where the duct from the pancreas enters. If you look at the histological structure of the pancreas, what does it remind you of? I heard some hissing very much. Yes, salivary glands. It's another one of these examples of a bunch of grapes. So the major part of the pancreas is made up of these acinar cells, which produce pancreatic enzymes. It's not exactly the same as the salivary glands, but the general structure is. And then there's a system of small ducts, which join with other small ones, bigger ones, eventually going into the duodenum. And these duct cells, in green here, produce a bicarbonate-rich mixture. This portion of the pancreas, which is forms about 98% of the pancreas, is the exocrine portion of the pancreas. And that's the only one I'm going to be talking about now. However, that other 2%, which is the endocrine portion, is of course extremely important. And this is made up of cells of the Langerhans, islets of Langerhans rather, which produce insulin, etc. And Dr. Harvey will be talking a lot about these later on in the course. So I'm going to just tell you that they're extremely important, but we'll be focusing on the exocrine. Exocrine meaning that the secretion goes to a system of ducts, endocrine that it doesn't, it goes out into the blood or extracellular fluid. So what are the functions of the pancreas? What does it do? Usual questions. How is it regulated? And how does all this tie together with the So here's just some facts. The pancreatic juice, isotonic, unlike saliva, which is always high-po. Most solutions are isotonic. It's alkaline, which is important, and 1 or 2 liters a day. We've seen that it produces bicarbonate. Other electrolytes too. Bicarbonate is the striking one. It's present in high concentrations. Inverse relationship between bicarbonate and fluoride.